Hi there, welcome back to the second half of the reproductive system lecture for Anatomy and Physiology 242. Oops, I realize I forgot to bring up Epic pens. Let me do that really fast. And then we'll get started with the rest of the presentation. So let's go full screen and we're back. Awesome. All right, so I'm actually backing up a little bit from where we ended in the Zoom lecture video I made, and that's because I want to remind that although we typically think of the production of gametes, sperm and eggs, as being meiosis exclusively, it actually does begin with mitosis in both cases, so both in oogenesis and spermatogenesis. We're looking at spermatogenesis right now, so let's focus our attention there. Later, we will compare and contrast oogenesis and spermatogenesis because there's some similarities, but also some pretty big differences. So, spermatogonia undergo mitosis, and I can't stress this enough. So, the difference between spermatogonia and spermatocytes is that spermatogonia are stem cells, and when they divide, they replace themselves as well as giving rise to future gametes. So they do both things. And that's why males have a different fertility window from females, specifically because of the persistence of stem cells into adolescence and adulthood, where people with ovaries don't have that. All right, so because spermatogonia undergo mitosis, the result is, by definition, two genetically identical daughter cells. Now, I want to stress, genetically identical refers to, of course, the nuclear DNA. It doesn't mean they have the same cell fate. So one of them is going to remain at the basement membrane and stay a spermatogonium. The other is going to differentiate into a spermatocyte and begin the process of meiosis. So that's the major difference there. So primary spermatocytes are the ones that begin meiosis, and the result of that is 4 non-identical gametes. So this is the mixing and shuffling of the genome. So here, right in this part of the image, this is the important bit, you get the formation of tetrads, and these are where homologous chromosomes that are duplicated. So you can see we go from one unduplicated copy of gray, which is homologous to black. Now we have duplicated homologous chromosomes, and these are going to trade bits. So maybe, for example, gray trades part of a leg with black there. So because of the shuffling, and also because of independent assortment, if you don't remember what those things are, go back and look at the information for Biology 160. Because of that shuffling and so forth, we're going to end up with 4 gametes that are not identical to each other. So they are genetically unique. So gametes possess a single set of DNA instead of a double, and we call this haploid. So what I mean by single set is each of us in our chromosome pairs have a pair of chromosomes in which one of our chromosomes came from our mom and one of our chromosomes came from our dad. So we are represented 50-50 by both parents. If you divide that, and due to independent assortment, maybe like gamete number 1 has like 45% dad chromosomes and the remainder is mom or something like that. Typically it shakes out to be 50-50, but which 50 isn't consistent across gametes. So single set of DNA just means a 50% amount with variegated mixture of parental DNA. This is why, for example, like, although I'm genetically similar to my brother, we're not identical because we got different sort of shuffled decks of genetic cards. Single just means half of a somatic cell. Another thing I want to stress before we move on from this topic is haploid. So one thing I notice when I teach Biology 160 is students fall into this verbiage of calling these cells haploids. That doesn't really mean anything by itself. So haploid is an adjective. It describes something. It's not a noun. So don't call things haploids. It has functionally no meaning. It just means a cell that is haploid. And those can be present for a variety of reasons. We use them to talk about gametes, but there are others. So do not use that nomenclature. It is incorrect. Okay, so just to kind of compare and contrast, in mitosis, you've got these chromosomes, and then before the cell duplicates itself, it duplicates the chromosomes. And then each chromosome, so let's see, the black one splits in half, and each cell gets one black one and one gray one, and so on and so forth. You get the idea. So that the chromosomes of the daughter cells equal each other. They're identical. During meiosis, due to synapsis, tetrad formation, and independent assortment, you get all of these different combos. So genetically unique gametes here. Okay, so this is a chart that I definitely need you to be familiar with because it's really critical for understanding and showing me that you understand spermatogenesis. So one idea that is central to this is understanding that spermatogonia undergo mitosis, and that both spermatogonia and primary spermatocytes are diploid. So those are going to duplicate their chromosomes and form tetrads. We don't call it anything right now. It's still considered to be diploid. The chromosomes are just in a duplicated state. And then we get secondary spermatocytes. And finally, when they differentiate into spermatids, then they are haploid. So spermatid is the noun for the name of these things. Haploid is their condition. So do not ever call these things here haploids. That's not correct. The reason I'm saying it so much is because people will probably still do it, but I'm trying. Okay, so these cells and these cells are genetically identical. It's just that they have different shapes from each other. So spermatogenesis is everything from here to here. Spermiogenesis is very different. So that is when the spermatids undergo some morphological changes, which is just a shape changes, and they become these little like light bulb shaped guys. And these ones are capable of swimming. So spermatids by themselves are not capable of motility. Spermatozoa, however, are. Okay, so let's talk about the function of nurse cells, and let's also write the other names for them up here. So nurse cells / Sertoli cells / sustentacular cells, and so on and so forth. So what these do is provide a barrier between the blood and the testis, and you can actually see that right here. So the spermatogonia stay below the blood-testis barrier, and the spermatocytes cross it and go into the lumen. So that allows for nourishment of the developing sperm, but it protects them from toxins and other bad things that would be in the blood. Another function of those cells is support mitosis and meiosis. So mitosis and meiosis are very ATP-consuming processes, and the nurse cells basically provide support for that. Another thing that they do is they secrete inhibin. So if you've been wondering about inhibin since we started talking about the endocrine system, now you know that inhibin feeds back on the anterior pituitary to inhibit the secretion of follicle-stimulating hormone. So we'll talk more about those feedback loops in detail. They are a little bit confusing, I acknowledge, more confusing than the regular like HP blah axis feedback loops, but actually less confusing in people with testes than in people with ovaries and a uterus, because like the uterus brings a third factor into the mix that's not present in people with testes. The other thing they do is they secrete a protein called androgen binding protein, or APB. And this specifically maintains high interstitial fluid concentration, that's what these square brackets mean, of androgen around cells, mostly testosterone. So it keeps T in the neighborhood to keep spurring on the process of spermatogenesis. Another thing that they do is they secrete something called Mullerian inhibiting factor. That's what this stands for. And this causes the regression of the Mullerian duct in the fetus. So if there isn't enough, you end up with, retention of parts of that tract, and that tends to inhibit testicular descent. And that can result in a condition at birth called cryptorchidism, which just means hidden testes or hidden testicles. So in some cases that can be corrected by surgery, but it's a spectrum disorder, so in other cases it results in infertility. It just depends on the severity. Okay, so here are some spermatozoa. The single term is spermatozoon. Again, this is another place in Latin where like you pronounce the two O's separately, so this word is not pronounced spermatozoon at all. It's pronounced spermatozoon. So the spermatozoa each have an head, and that's of course the obviously head portion, containing acrosomal enzymes. So the acrosome is a membrane-enclosed lysosome-like structure that sits on top of the nucleus. So the goal is that when this spermatozoon makes initial contact with an egg, that acrosome bursts, and that helps the sperm burrow through the zona pellucida and gain access to the outer surface of the ovum, which is supposed to penetrate. The middle piece is also called the shaft or rotor shaft, and this has mitochondria in it arranged in a spiral. So if I were to draw this a little bit bigger, you would see the mid piece looks like this, and then let's draw ourselves ahead. And then the mitochondria are kind of situated around the central axoneme of the microtubule that supports the flagellum in sort of a barber pole-like fashion, which is pretty nifty to look at. And then, of course, the flagellum itself. So that is the swimmy tail part that allows the sperm to move and propel itself forward. So these are highly specialized cells. They are not generalists, which is why they are so pared down. So like they're you know how there's a difference between like a general purpose vehicle, like a sedan or a station wagon, where you can do lots of different stuff with that car, but you know, at the end of the day, it's a car that goes, versus a very specific sports car like a McLaren or something that's like, it's a car that goes, but like it's not really a grocery getter or a kid carrier or what have you. The sperm is more like the sports car, so it has a very specific purpose, and that is to find an egg and hopefully go faster than all the other sperm. So these are for DNA delivery only. They don't do anything else, and they are powered by fructose. And then this fructose concept will come back in more detail later when we talk about the process of producing semen, which is actually quite involved. There's a lot of steps. So here we have the male reproductive tract and two views of it. So notice that the penis is anterior and hangs underneath the pubic symphysis, but the root of the penis is actually situated back here, back here deep in the pelvis. So it extends considerably further back than its apparent length. The testes and epididymis, of course, hang from the body in the scrotum, which allows for thermoregulation. And then in order to get out, sperm must be propelled up through the ductus deferens over the top of the bladder, where they link up with the seminal vesicle to form this part of tubing called the ejaculatory duct. That joins up with the prostatic urethra, which is the part of the urethra that goes through the prostate gland. And then after that, it becomes the membranous urethra, which is right here only. And then any part of the urethra that's in the penis is called the penile urethra. So that's what's there. Oh, here's the bulbourethral glands as well. So we're going to discuss the production of semen and how sperm eventually make it out of the penis. So the general features that I just showed you are for the following purposes. The spermatozoa, they're relatively fragile cells. So they need a lot of help and support, and that's because they don't have a lot of cytoplasm. They create energy for a very specific purpose, and that is to swim. And so they need maturation, storage, and transport. And those things are all provided by the male reproductive tract. So that's the epididymis, the ductus deferens, the urethra, and the glands that I mentioned. So let's go through all of those. The epididymis is this comma-shaped organ that sits on top of each testis. So if the testis is where sperm are made, the epididymis is sperm finishing school. So they're stored in there, they hang out in there, they learn to swim in there, they do all kinds of things. If you were to uncurl it, it's about 23 feet long. How do we know that? Someone did it. Every time you see one of these weird length metrics, it's because somebody bothered to measure the length of the epididymis or some other thing. So no one's making these figures up. Somebody actually went ahead and unfurled an epididymis and measured it. These are things you can get scientific funding for, miraculously. So this absorbs fluid arriving from the seminiferous tubules. And I want to stress, absorb isn't quite the right word here because that implies that the fluid passes into the wall of the tube somehow. It doesn't. It's just that the fluid containing spermatozoa flows from the testis through the reedy testis into the tubules of the epididymis. So this is a PowerPoint that I adapted from somebody else. This is not, absorb isn't the word I would have chosen, but it's here, so I thought I would address that. So just don't be thinking that the fluid is crossing from the lumen into the epithelium of the epididymis. That's not what's happening. Another thing this does is recycles damaged spermatozoa. So similar to protein synthesis, spermatogenesis and spermiogenesis doesn't always go perfectly. Sometimes you get a janky sperm that's not fit to go try and reproduce. So those need to be taken care of, and that's one of the functions of the epididymis. So this is capable of storing and protecting spermatozoa for about 2 weeks. And during this time, the spermatozoa are mature and capable of swimming, but they're not moving much, and they're certainly not swimming around. They may twitch, but that's about it. Okay, capacitation. So this is a process that spermatozoa undergo. So during capacitation, basically it's the sperm waking up. So the spermatozoa become motile and functional. So they get the ability to swim and do their job, which is, of course, DNA delivery. So one of the requirements for capacitation is that the spermatozoa be exposed to seminal secretions. So these secretions have eicosanoids like prostaglandins and other cytokines that actually sort of spur the sperm to wake up and begin to get ready to do its job. And the conditions in the female reproductive tract also facilitate capacitation. So the ductus, or vas deferens, extends from the epididymis to the ejaculatory duct. And it takes this really long path. So remember when I was talking about the spermatic cord passing through the inguinal canal and how that predisposes people to inguinal hernias? You can see that here. So let me make my little arrow smaller. So it has to go up through this hole right here and then pass over the top of the bladder. So it doesn't go like this, which would make more sense linearly. It goes up and around, and that's due to the way that this structure develops. So here's the posterior view of the urinary bladder. And you can see that the ureter, which carries urine from the kidneys down to the bladder, is crossed over by the ductus deferens. And then the ductus deferens widens before it joins up with the seminal vesicle, and that area of the ductus deferens is called the ampulla. So it's got a very thick wall of smooth muscle, which you guys have already seen in the histology video I posted, but here's a scanning electron micrograph of it. So this is all smooth muscle. And that should tell you that the spermatozoa, as they pass through the ductus deferens, are not doing so because they are swimming. They are doing so because they are being propelled along by that smooth muscle. So we call that peristalsis. And the ductus deferens can also store spermatozoa. So as long as, as long as FSH and LH are being secreted with regularity by the anterior pituitary, spermatogenesis will proceed in the testes, which means that you must provide or make room for older sperm to be stored if they're not going to come out anytime soon. So both the epididymis and the ductus deferens serve that purpose. Sperm storage between ejaculations. Okay, the urethra. So the urethra is basically divided into a couple sections. In total, it extends from the base of the bladder, so right here, to the tip of the penis, which is right there. And there are sections of the urethra. So the prostatic urethra, let's make them different colors. So I'm going to make the prostatic urethra blue. And then the membranous urethra, which is orange. It's just this little part here. That's actually not a good enough color for seeing. It's just this little part here that passes through the pelvic floor. And then the penile urethra, which is the rest of this, all the stuff that passes through the erectile tissues of the penis. So those are named because urologically, so if you're a urologist, you study pelvic contents and urinary health, different parts of the penile urethra and the other parts, prostatic and spongy, or excuse me, membranous, are subject to different ailments. For example, an early sign of prostate cancer is difficulty voiding the bladder because the prostatic urethra gets kind of smooshed shut. And that creates difficult and painful urination for people that suffer from that. So the accessory glands in the male pelvis are as follows. And let's go through them one by one. So they contribute 95% of the volume of semen. So really, when a semen sample is taken either to assess fertility or for in vitro fertilization or some such, most of what is there in the sample are not spermatozoa. It's other stuff. So this consists of the seminal glands, also called the seminal vesicles. The prostate gland, which I've just described a little bit, and the bulbourethral glands. And each of these glands contributes their own fluid, and each of these fluids has different constituents, but all of the fluids constituents basically help to make sure that the sperm are assisted in hopefully completing their task of fertilizing an egg. So the secretions of the seminal gland, or seminal vesicles, consist of a variety of different things, and we'll go through them because each of them have a very specific function. These also contribute the largest amount of fluid volume to the semen. So the concentration of fructose in the seminal vesicle secretions is high, and this is of course to nourish the spermatozoa. So let's write I also mentioned that capacitation involves signaling molecules that help to ensure that the sperm are sort of woken up as they begin to make their way out of the reproductive tract. And one of those signaling factors are prostaglandins. So remember from endocrine system, these are eicosanoids. And in addition to helping with capacitation, prostaglandins and other signaling eicosanoids stimulate peristalsis both in the male reproductive tract but also in the female. So they're providing muscular action that assists in getting the sperm where it needs to go. Fibrinogen also occurs for semen clotting. Now, why would you want semen to clot? That seems kind of icky, right? If you think about it from a reproductive standpoint rather than a silly human standpoint, it actually makes a lot of sense. So, I'm going to switch over to board work here for a minute to kind of explain why, because it's an important piece of reproductive function, and it's an interesting adaptation, actually. I want to remind you, we're going to talk about fibrinogen quite a lot when we talk about blood next. So I'm talking about the same fibrinogen that is involved in clotting the blood. So both apply here. Okay, so let's first draw a female reproductive tract. And I'm going to make it, I don't know, green for fun. Why not? Things can be whatever color. We don't have to stick to gender binary colors at all. So here is a uterus. Here's a cervix. And then here's the vagina. So let's label that. Okay, so let's assume I'll have like a fallopian. Let's do this. Here's an ovary, and then here's a little fallopian tube coming off from the side. And I'm not going to draw the other one. So if the goal is to get spermatozoa here, and if you're thinking, shouldn't the goal be to get the spermatozoa into the uterus? The answer is no, actually. So most fertilization events that are successful, and indeed most fertilization events in general, actually happen in the oviduct. So fertilization takes place in the oviduct, and then the product of conception, so the zygote and subsequent stages of embryonic development, those make their way down the rest of the oviduct to settle in the uterus. So this is why sperm's job is so hard. They have to get into the vagina, hopefully find their way through the cervix into the uterus, and then they have to find the oviduct, and also they have to find the correct oviduct. So, you know, if like half of the sperm go left and half of the sperm go right, and it was the left side that ovulated that month, that means that all of the sperm that are going right are on a fool's errand. They're going to die for no reason. So, in fact, the odds of each of us individually being born as we are, so like as the genetically unique individuals that we are, statistically is pretty low. So I hope that makes you feel special today. Makes me feel special. All right, so here's the goal. Here's why you might want to have semen clot. So let's make the vagina a little longer. Usually the vagina is about 4 inches long, shorter than most people think. All right, so there's the vagina. Let's pick a color for penis. How about orange? Yeah, why not? So, during ejaculation, here's what happens. Semen is expelled here, and then withdrawal usually occurs. So you get the penis being withdrawn from the vagina, sex is over, right? If, like, let's say the person with the vagina and the uterus is like, well, okay, I have stuff to do. I'm going to get up and leave the sexual encounter. If that was the case and there was no fibrinogen, the semen would just fall back down immediately, and that would impair the ability for pregnancy to occur. So the fibrinogen helps to clot the semen around the cervix, and that helps give the semen time, or the sperm time, to try and make it up into the uterus and subsequently the oviduct. So let's add this So it contains fibrinogen to clot semen and hold it near cervix for a bit. And then hopefully that will give the spermatozoa just like a little bit of an extra leg up or an assist in trying to reach what they're supposed to be reaching, which is, of course, the oviduct. I'm actually pretty proud of this drawing. It looks like a fun anatomical neon sign. Let's give it a label. Ta-da! Awesome. I'm going to take a picture of that. I'm proud of it. Okie doke. Goodbye drawing. Let's go back to business as usual. So one point I want to make, and this is an important one because a lot of folks fail to realize this, is that emission and ejaculation are different things. So emission happens prior to ejaculation, and that is the process of making sperm. So a batch of semen is whipped up during intercourse as ejaculation is prepared for. So it's not like you have all these secretions pre-existing ready to go. They are added as semen arrives in the ejaculatory duct right before the moment of ejaculation. So that's what emission is. This is the, let's call it the construction of complete semen prior to ejaculation. So that's what emission is. So the seminal vesicles have obviously a strong role in emission, but they are by no means the only player in this game. So let's go look at who else is involved. Oh, one more thing before we go is that not surprisingly, this process, so emission in general and emission specifically to the seminal glands, is dominated by sympathetic activation. There are some components that are parasympathetic, and we'll address those, but remember, sympathetic is fight or flight, or for responses of the human body that need to be brief, intense, and immediate, and emission and ejaculation definitely qualify, so it's not surprising that it's involved. Okay, the prostate gland. So this is sort of a plum-shaped gland, and it sits at the base of the bladder, and a bunch of tubes pass through it. So the ejaculatory duct passes through it, as you can see. This contributes another 20 to 30% of semen volume. It varies a little bit, and it has some other functions in addition to those of the secretions of the seminal vesicle. So let's talk about them. One is seminal plasmin, and this is an antibiotic. So one thing you don't want is for your sperm quality to be compromised by bacteria. And bacteria love warm, dark, moist places. The interior of the reproductive tract of the male and the female both qualify as that. So it's unreasonable to suppose that it's a sterile environment. There are bacteria. In fact, there's more bacteria in the female reproductive tract because there are commensal bacteria that help keep the vagina healthy, for example. So in order to avoid spermatozoa being interfered with by those bacteria, seminal plasmin is basically there to resist antibiotic or biotic infection. There's also a clotting enzyme that activates fibrinogen, so let's add that. And there's also proteases. So there's also fibrinolysin, and this is going to dissolve the clot after it's served its purpose, and also dissolve the mucus after it has served its purpose. The prostate gland is also under sympathetic activation. So it only contributes its fluids right before ejaculation, so during emission as well. Okay, bulbourethral or Cowper's glands. So these are paired. They're on either side of the base of the penis. In fact, let's go back. So I want to point out that from the back view, you can see one on each side. And they both have these little openings into the membranous urethra. So these Cowper's glands are situated in this picture here, so you can only see one of them on one side of the penis. So these are the exception to that sympathetic rule. These are undergoing parasympathetic activation. And they produce a thick, clear alkaline mucus. Then that is during the arousal phase. So this is often referred to as pre-ejaculate. And what this helps to do is the following. The penile urethra is a dual-purpose tube, so it carries both urine and semen. It has to do both, not at the same time, but semen, and specifically spermatozoa, is really, really vulnerable to acidity, and urine is often acidic, and contains waste products that would be harmful to spermatozoa. So prior to the passage of spermatozoa through the penis and out of the body, the penis prepares itself by sending this thick mucus through the alkaline mucus. The alkaline portion basically functions to neutralize any acid that might be there. It also clears any debris out of the urethra, and it aids in intromission. Intromission is the scientific word for the act of inserting the penis into the vagina. It lubricates the glans penis, which is this end part here, and that helps to facilitate intromission. So this is under parasympathetic activation. Okay. So semen itself is all of the things I just described. The typical volume for ejaculate is between 2 and 5 milliliters, and this contains a lot of spermatozoa, so even though it seems like a small volume, it's definitely enough. I mean, you and I and everyone we know, we're all born, weren't we? So clearly it's good enough. It is slightly alkaline to oppose the acidic conditions inside of the vaginal canal, and its pH is about 7.2, so just a little bit north of neutral, not even as alkaline as blood. So what's in it? Well, spermatozoa, and that's between 20 and 100 million, depending. So what does it depend on? Depends on hormone levels and testis health, right? So for example, if somebody's getting their fertility assessed and they have a low sperm count, one of the questions that a clinician might ask is, well, let's do some blood work and see if you have low steroid hormones, so maybe low testosterone, or perhaps low FSH, LH, or GnRH. That would be the endocrine line of questioning that a fertility specialist would go down in response to that finding. Okay, so seminal fluid, what does it contain? Seminal gland, prostate gland, nurse cells, epididymis, and bulbourethral gland secretion. So nurse cells undergo apoptosis and are shed into the lumen sometimes, which is why if you look closely at the histology guide slide, for example, you can sometimes see shed nurse cells in there. And then there's also enzymes, including proteases, seminal plasmin, that's that antibiotic one, clotting enzymes, and fibrinolysin. So with regard to the external genitalia, you have the scrotum, which is derived from the urogenital fold of the embryo. And then you have the penis, which consists of the root, or cru, that's how that is pronounced. And then the bulb is attached to the corpus spongiosum. So the cru are attached respectively to each of the corpora cavernosa, and then the bulb is attached at the base of the shaft, which is the corpus spongiosum. The glans is the expanded end of the corpus spongiosum. At birth, all penises are covered with what is called a prepuce or foreskin. And this helps to protect the glands, A, from drying out and toughening, but also B, from things like abrasion and reductions in sensitivity due to constant rubbing on clothing. And evolutionarily, that's a relatively new phenomenon because, of course, this whole system evolved prior to the advent of clothing oneself. So if you're imagining proto-hominids as wearing underwear, stop, they didn't. All right, so the penis in cross-section looks like this. Kind of looks like a little surprised alien guy, almost like E.T. And that is because of the way the erectile tissues are shaped and arranged. So it's an elastic connective tissue, so it's a mixture of, let's write this down, mixture of dense irregular and elastic fibers. And it's got a lot of holes in it. And each of these structures, so 1, 2, the corpora cavernosa, have a deep artery. So that's how the blood gets in during the process by which an erection occurs. So what creates the turgidity? Turgid means rigid or stiff. What creates the turgidity is that blood can get into the sinuses of the corpora cavernosa through that central artery, but the veins are constricted so it can't get out. And because the tunica albuginea is going to not allow the expansion elastically of the penis, that it becomes stiff instead of getting bigger. So it doesn't swell like a blood swelling, it becomes turgid. Come on, I would very much like to advance the slide, please. Thank you. There we go. So the corpus spongiosum also undergoes erection, but not to the same extent that the corpus cavernosum or corpora cavernosa, plural, does. And that's because the spongy urethra passes through it. If it got too stiff, it would close the urethra, and then that would deny the passage of sperm out of the penis. So that would be bad, right? Okay, so here is the dorsal artery. So during arousal phase, one of the things that happen is that nitric oxide, remember NO, so nitric oxide is a gasotransmitter, so gaseous neurotransmitter. And specifically, it causes smooth muscle. Wow, I can't write today. Smooth muscle. There we go. Relaxation and dilation. So what this means is the arteries that are admitting blood, so this deep artery here, let's outline it in red. So this guy and these guys are going to dilate and become very wide, and that permits blood to enter these sinusoidal spaces. So this is going to happen during arousal, and that is what creates turgidity. It also compresses outflow because the blood is pushing against all this, making these vessels very small because they're occluded, they're thin, and it also compresses the dorsal vein, which makes blood exiting difficult. Okay, so this is the slide that we're going to stop on, but of course I'll explain it first. I'm not just going to leave you hanging. And notice there are some question marks here, and that's because many parts of this feedback loop are pretty well understood, but not all of them. So there's still some mystery in here, so if you decide this really interests you and you want to go do a PhD about it, you could find out the question mark if you want. Okay. So we've got release of gonadotropin-releasing hormone coming from the hypothalamus. And that is going to stimulate follicle stimulating hormone secretion and luteinizing hormone secretion. So as I mentioned, FSH is going to basically stimulate the nurse cells to promote spermatogenesis and spermiogenesis, and also secrete androgen-binding protein and inhibin. So inhibin feeds back on the anterior pituitary to stop its release. It might also feed back on the hypothalamus, but the question mark is there because, of course, no one has verified that for absolutely sure yet. So the effect here is when you get an increase in androgen-binding protein synthesis, testosterone sticks around the seminiferous tubules more, and that helps promote the differentiation of sperm. And then the nurse cells themselves just continue to stimulate spermatogenesis and spermiogenesis. Luteinizing hormone acts on the interstitial cells of the testes to cause testosterone production. So one thing that is very clear is that testosterone feeds back on the hypothalamus quite strongly. So the hypothalamus is known to express testosterone receptors, and testosterone inhibits the secretion of GnRH. The peripheral effects of T are maintaining appetitive sexual behavior, so that's sex drive. Appetitive means the desire or motivation to do something, and that's coordinated by very specific parts of the brain and hypothalamus, just so you know. It also stimulates bone and muscle growth, and establishment of male secondary sex characteristics. So androgens, including dihydrotestosterone, that's the big skin one. That's what, for example, changes the facial skin of young males from being hairless to being hairy with the onset of puberty. And it also maintains the accessory glands and organs of the male reproductive system. So a lot's going on here. All right, and that is where we're going to stop for reproductive system for now. I would like to have finished this PowerPoint, but it is 88 slides long, so that's not really feasible for one video. So now I'm going to go ahead and get to work posting this for you, and I will see everybody on Zoom in a few minutes.